Hi, everyone. My name is Kenny Coogan. I'm the Education Director for the International Carnivorous Plant Society, and welcome to another exciting webinar. We started these in January of 2022. We used to do one a month, but this month we have two, and next month we have two, and the following month we have two, which is one of the benefits of being a member of ICPS. Today, we're going to be talking about Sarracenia alata, the effects of prey capture and pollen-donor distance on seed production by Hali'a Eastburn. But first, I wanted to remind you that we have monthly happy hours where you can bring a drink to your computer and a plant and share your knowledge and ask questions. And we always have a really fun time with that. And as you can see, we have a bunch of webinars scheduled, and we also have a few that I haven't posted yet. If you visit our Facebook page, you can see all of the events, or if you're a member, you can go to Club Express and you can get the Zoom links and the description of all of the events. Another exciting thing we've been doing for the past 2 years is World Carnivorous Plant Day. If you go to our Facebook page or our YouTube channel, you can see over 50 videos of professionals, hobbyists, growers from all over the world celebrating carnivorous plants. We talk about conservation and cultivation, and you can check out all those archived videos there. And part of World's Carnivorous Plant Day every year is we have a photo contest. You can submit your photos from January to April, and you can submit a bunch of photos. And if you're one of the 3 winners, you get a 1-year membership to ICPS, and it's free to enter. And World's Carnivorous Plant Day is the first Wednesday of May, so that means it changes every year. We wanted to do it in May because a lot of members are from the Northern Hemisphere. We didn't want to do the first Saturday in May because that is World Naked Gardening Day, and that would be a dangerous combination. So next year, May 2023, World's Carnivorous Plant Day is going to be on Wednesday, May 3rd. Usually we post one video every hour for about 24, 26 hours, so there's lots of content. You can support World's Carnivorous Plant Day and the International Carnivorous Plant Society by purchasing some merchandise. The proceeds from that goes to our Carnivores in the Classroom grant. Last year, we were able to fund 24 classrooms, $150 each, to add carnivorous plants to their classrooms. And teachers, K through 12, applied from all over the world, and we were very happy with that. Here are some photos. And right now, it's August 9th. The month of August is when teachers can apply. So August 1st to August 31st, the grant period is open. So if you have teacher friends, please tell them. This is going to be going on YouTube in a few days. Another thing is, for the month of August, we partnered up with the Carnivorous Plant Club, and we have a little fundraiser going on. Our goal is to get $10,000. Half of it would be for conservation, and the other half would be to further our education ideas and videos. And without further ado, we're going to have our main guest. If you are attending live, please type in your questions in the chat box, and I will ask her at the end. And all right, Hali'a, you are the host now.
All right.
And we are very happy to have you here. We're excited to learn about your thesis.
Thank you for the invite. This is great. I love being able to share what I accomplished over a couple years studying these really extant populations of Sarracenia alata that are present here in Texas. Dr. John Horner was my major advisor at Texas Christian University. He's been working on Sarracenia alata here in Texas for about 12 years. And my project is one of very many trying to characterize this population and really understand the many factors that are impacting these remnant populations and what we can maybe do to preserve them and enrich them even. And so my project was looking at the impacts of pollen donor distance and nutrient availability on the reproductive success in Sarracenia alata, a carnivorous plant. So a very prevalent theme in biology is the variable nature of environments and how those affects ecosystems, populations, and even individuals. Environmental changes can be biotic or abiotic, and they might look like human-initiated or natural climate change. They could also look like predation, parasitism, or attacks from other enemies. But across all taxa, fungi, plants, animals, bacteria even have all come up with a strategy to survive in the face of a changing environment, and that is sexual reproduction. So most organisms benefit from having diverse genetics. It ensures that their offspring have an increased probability of having variable genetics that are going to be suitable in a variable environment. However, there are many factors that impact the success of this survival mechanism. The first being the genetic relatedness between mates, and secondly, the nutrients that are available for the physiological processes of sex, such as creating pollen and seeds and everything like that, that is very energy intensive. So there are distinct disadvantages to being too closely related or too distantly related to your mate. With inbred mating, we see an increased frequency of deleterious recessive alleles. And with outbred matings, we see a loss of local adaptations that might be advantageous to these offspring. So most organisms have some kind of mechanism, whether it's behavioral or physiological, that will avoid these two extremes and select for an optimal difference between mates. In plants, because they're sessile, meaning they don't move, we can correlate genetic relatedness to the physical distance between pollen donor and pollen recipient. So we want to know, what is the effect of genetics on reproductive success? That's a very basic question in plants, and it's one that we start with here. So we might find in our population, because it might differ population by population, that as pollen donor distance increases, reproductive success increases as well. Perhaps you have some inbreeding depression here, but it continues to increase as your pollen donor distance increases as well. Or we might find that we'll have inbreeding depression here, and as pollen donor distance increases, we get outbreeding depression on the other side, where we're losing those localized adaptations, and we have this optimal degree of difference here in the middle. Nutrients are super important for reproduction. They are the energy that's required to produce gametes. In plants, that means pollen, seeds, ovaries, even flowers. And plants source nutrients directly from their environment. And these are a limited resource, and they're differentially allocated. And so deficiencies in nutrients can lead to a lot of problems: reduced growth, reduced reproductive capacity, or a combination of both of those things. Generally in plants, we expect to see this relationship between nutrients and reproductive success. As nutrients increase, we get better reproductive success. Pretty straightforward. However, nothing works in isolation in nature. So we are always interested in interactions between different factors, here being genetic relatedness between mates and the nutrients available for reproductive success. And there's a few ways this could go down. We could find that with increased pollen donor distance, regardless of nutrients available, we're going to have increased reproductive success. And we might also find that increased nutrients aren't going to necessarily help with plants that already have inbred matings or anything like that and have naturally poor genetics. But they're going to be a real big help if you have great genetics as a result of outbreeding. So this is a couple possibilities, and we wanted to see what our population was going to show us. Across the literature in flowering plants, there's variable results as far as what this interaction might look like. Some of the research showed no effect of this interaction between genetics and nutrients. Some showed increase in breeding depression or decrease in breeding depression. It was really all over the place. But what these studies all had in common was that none of them were actually actively restricting nutrients. What they were doing was comparing baseline field conditions with a surplus nutrient situation, which in reality is not really going to happen. I guess unless you're over fertilizing your fields or something like that. But in a natural situation, and unfortunately with carnivorous plants, we're never going to have a huge abundance of nutrients that are naturally going to be in the environment. And so we wanted to look at a more real world situation and approach our experiment that way. So we approached this by using the carnivorous pitcher plant Sarracenia alata as a model to study the relationship between genetic relatedness and nutrient availability in flowering plants. And Sarracenia flowers arise early in the season before the carnivorous traps open and become active. And they are pollinated primarily by insects. The bumblebee is their main pollinator. And again, these blooms are going to emerge first thing in the season before the traps open. And Sarracenia is found in nutrient-limited bogs, and it gets majority of nutrients from insect capture. And these modified leaves here are called pitchers. They lure and capture insects and produce endogenous enzymes, which help to break down these insect prey. They also host a variety of very specialized fungi and bacteria that also help with nutrient absorption. The pitchers become active after pollination is completed, and this helps the plant from devouring its own pollinator, which again is the bumblebee. This temporal separation is also great for us as researchers because we are able to manipulate both pollination and this feeding mechanism separately without interacting or without disrupting either process. And because of this special nature of Sarracenia, we are able to actually create a nutrient deficit that is very true. And so the previous work in nutrient availability in Sarracenia is really sparse. Most of the work was done in the early 2000s by Jessica Butler and Aaron Ellison. And in their studies of Sarracenia purpurea, they found some really interesting things. The first picture of the season that Sarracenia puts out is created majority from stored nitrogen reserves from previous seasons. And this thin shaded band represents the proportion of this picture that is constructed from current season prey capture. So as we get subsequent pictures arising throughout the season, more and more of the picture is constructed from current season prey capture. And what is missing though from the study is that nutrient cycling does not include the energy that's needed for reproductive organs, which again are put up first thing in the season. And so our hypothesis going into this study was that pitchers are contributing current season nutrients to seed filling, much like they are to construction of their pitchers, because seed filling is a process that starts at the beginning of the season, but it goes completely through the entire growing season up until harvest time, which is in October. And so we should be able to see some of that impact from that nutrient contribution. This map shows the counties where there are known Sarracenia populations in its range. And this star represents Leon County, which is where our study site is located in eastern Texas. Now, Sarracenia alata is populations in Texas are extremely fragmented and extremely sparse. We have records of them being as far north and west as Fort Worth, But today, these are the only remnant populations that we have left. And this is the only pitcher plant that is native to Texas. Genetic studies done by Dr. Horner and others have shown that this species was once contiguous across its range from East Texas all the way into Alabama. And today it's not federally listed or protected because of these very locally abundant populations in Mississippi and Louisiana. So that doesn't help us with protection of these extant populations out here in Texas. This is our study site. It's located on private property. And the nearest pitcher plant bog is approximately 16 to 25 kilometers away. That's 10 to 15 miles. And again, their pollinator is bumblebees, and bumblebees only have a foraging range of about 2 kilometers or 1 mile. So that means that our population is reproductively isolated, meaning that individuals in this bog are only reproducing with other individuals in the same bog. This makes it super important that we understand the status of reproduction in this population because it's so characteristic of the other fragmented populations that we find here in Texas and other fragmented populations of Sarracenia across their entire range. Pitcher plants also have very unique relationships with insects, fungi, and bacteria. And they represent another layer of biodiversity in these ecosystems, which are considered critically endangered after nearly 400 years of land conversion, urban sprawl, and draining of these wetland ecosystems that they are a part of. So we had a few questions going into the study. We wanted to understand if there was an optimal distance between mates in our population. We wanted to know if there was an interaction between genetic relatedness and nutrients available to these plants and how that impacted their reproductive success. And then, as always, we want to know how this information is going to help improve conservation of fragmented Sarracenia populations and plant populations in general. This is something that we're experiencing a lot of. And so this information can be used for a lot of other plant genuses. Before I go any further, I wanted to acknowledge the plethora of people that assisted me in this project, my thesis committee, Dr. John Horner, Dr. Dean Williams, and Dr. Amanda Hale from TCU, our bog site landowner who keeps really great care of this site. He does not understand a lot of the science and he's not like a big collector or enthusiast of carnivorous plants, but he knows he has something special there. And he takes really good care of that site, giving it prescribed burns and keeping it really, really nice for us to work on and study. My lab mates, graduate students, friends and family, and then of course, funding from CERC and ADKIN grants from TCU, and a big thank you to the Native Plant Society of Texas, who funded purchase of a lot of great grow lights that enabled us to do the first ever germination trials for this population. So we employed a cross-classified design and applied 2 different treatments to our study population. First, we hand-pollinated randomly selected flowers with pollen taken from donors from varying distances. 0 meters here represents selfing, and then we had 5 out-cross distances, 35 meters to 190 meters. And 190 meters is the entire stretch of the bog. And the second treatment we applied was prey exclusion. So half of our study plants were left open to regular prey capture for the entire growing season. The other half, we excluded prey completely from every single pitcher for the entire growing season. So this is some pictures of our site. At this particular site, it's a hillside bog that goes down into this little pond. And it's surrounded by longleaf pines. This side, it's open to a lot of sun exposure, and these plants are found in these little patches all over. And so we selected some nice sized patches to work with and get donor pollen from. And doing various crosses between each patch is how we got our 5 out-cross distances. All right. So starting in March, we put on these pollinator exclusion bags onto all of our randomly selected flowers. We secured these bags to these flags. Texas is notorious for crazy spring storms and tornadoes. And so we needed to make sure that our flowers were protected for the entirety of the season. As they started dropping pollen, which you can see here on the top right, we knew that they were receptive to pollination. So we began our hand pollination treatments. For our outcross distances, we took pollen from 7 to 10 donors at each donor distance, mixed that up, and applied that to selected flowers. And then for self flowers, they just got their own pollen. Plants, the bags were resecured to the flowers and left on for the entirety of the season to make sure no other insects could get in and pollinate after we did, or could get in and destroy our flowers. And then starting in about April, pitchers at this site began to arise and open up and become active for prey. And so on our selected plants, we excluded prey by putting a simple piece of cotton into the mouth of the pitcher. And then we did so on every single pitcher on our selected plants. So they had 0 prey capture for the entire season. We returned every 2 to 4 weeks to plug any new pitchers and to check the integrity of the site. We had a few deer move through the site, pulled flags down or flowers over, and it was kind of a mess, but it wasn't too bad. Historically, this site gets some hogs, like feral hogs that run through, and thankfully we didn't have any problems with hogs or anything crazy like that. So those prey exclusion treatments continued up until the beginning of October, which is when we harvested our ripened and mature seed pods. We collected a lot of data. We wanted to collect seed number and mass, ovule number and mass, and ovules are unfertilized seeds. And from that, we found the proportion of total ovules that were producing seeds. Here in this picture, you can see the nice fat seeds here on the left that are filled. And on the right, you can see these little tags are the ovules. And these are unfertilized seeds. They're still attached to the placenta. It's really interesting. And then we also wanted to get a handle on offspring fitness. And so to do that, we germinated a subset of every replicant that we produced. And we did these germination studies in Petri dishes with dampened filter paper. We didn't want to introduce any type of media that might provide an advantage to some seeds over others. So we just wanted to see raw potential. And seeds that were ungerminated at the end of this period, at the end of our 30-day period, we tested for viability using a tetrazolium chloride test. And this red dye here indicates that that seed is viable. And overall, I manipulated 200 free plants, collected 198 ovaries, analyzed them. We found an average of 940 seeds per ovary, which worked out to about 186,000 seeds, which I personally hand counted, and then germinated 3,360 seeds over 168 Petri dishes. A lot of data, a lot of seeds. It was pretty much my whole Christmas break. And what we found was that as pollen donor distance increased, we saw an increase in seed production, which was great. That's what we wanted to see. And so we see here total seed number on the y-axis and increased pollen donor distance and a nice steady slope up. However, we found no significant effect of nutrient availability or an interaction between nutrients and genetic relatedness on seed production. This graph shows proportion of total ovules producing seeds on the y-axis, and our two extreme pollen donor distances, 0, which is self, and 190 meters, which is our furthest distance possible within this bog. And it's very interesting actually to see that the pitchers that were blocked started out in the selfed area. They were actually producing a slightly higher amount of seeds compared to those that were open to prey capture. But when you get to the furthest distance, the seed production was about the same. And so that's a really interesting difference, though, and we're still thinking about that one. We also found no effect of nutrient availability or genetics on the proportion of viable seeds that were produced, so on our quality of offspring that were produced. And at first I was really frustrated because I wanted to see a difference, but it's actually super interesting because in a plant that is in a nutrient deficient environment that is so dependent on insect capture, for it to have no effect on any aspect of fitness in this sense was really interesting. So in science, we ask questions and we usually get questions back, and that's what happened here. And so we returned to the literature, and really our results align with previous studies done by Neiman et al. In 2006. And they found that seed production was really only affected when there was a 5-fold increase in nutrients. And they restricted photosynthesis, prey capture, and the combination of those. And then they did supplemental feedings. And it wasn't until they did the supplemental feedings that they saw an effect on seed production. And Jessica Butler in 2008 showed us that Sarracenia alata stores up to 25% of its assimilated nitrogen from a previous season to be used in the next season. And so we're wondering if seed production and flower construction is solely dependent on the stored nutrients, or perhaps the methods that we employed in this study are just not fine enough for us to see the amount of nitrogen that's being used in this current season. And that's totally reasonable as well. Another question we've got is, how does pitcher number influence nutrient cycling? So this returns us back to Butler and Ellison's work. And we see here that it's not until the 5th or 6th pitcher in Sarracenia purpurea that the majority of the pitcher is being constructed from current season feeding. And so perhaps if you have fewer pitchers, like if they only produce 1 or 2 or 3 pitchers, these pitchers are going to be acting as a nutrient sink rather than a nutrient source for construction of tissues, whether that's pitchers or seeds or flowers or anything like that. The field season, when we did this work, the average number of pitchers that we saw on each genet was 2 to 3 pitchers. That's really low. We've seen up to 20 pitchers per plant. And we're not quite sure why in this season we didn't get as many pitchers. It could have been an impact of prescribed burns or something that had happened in previous seasons. We're really unsure. And so that is something that we would like to pursue in the future, just understanding that relationship. This is some historical data collected by Horner Lab at TCU. And the proportion of ovules fertilized, so this is a metric of seed production, from 2008 to 2016 holds pretty steady at 75%. That's historically what we've seen. The abundance of pollinators falls in line with that seed production that we've estimated. And then in 2019 and 20, we had a huge decline in our seed production. It went down to an 11% seed production level. And those years, we also saw less pollinators in the area. And then back in 2021, which is the year of my study, seed production levels were up to 90%. There was bumblebees everywhere. And so there was a few factors that might have caused this. Perhaps chemical applications in the area. There's a lot of agricultural area around this particular bog, and no pesticides select just for bumblebees. And so it's likely that any kind of pesticide application was also damaging the insect prey populations as well. And so they might have been hit by low pollinators and low prey capture for 1 or more years. And this decline that we're seeing in 2019, 2020 might be a result of previous declines in those populations. Again, we don't know. We're really interested now in the natural fluctuations in insect capture and these populations. Is this a normal thing that happens for Sarracenia? Is this why they rely on stored reserves so well? Or is this an anomaly and something that we need to be worried about and careful about going into the future? So returning back to our questions, is there an optimal distance between mates? In our population, we can say that distance is greater than 35 meters will maximize reproductive output. We don't want patches. We don't want that bog to get any smaller than it is. 35 meters is really the smallest, and that probably applies to other Sarracenia populations. And we've seen here in Texas populations that are about 35 meters wide. That's how small they are at this point. And so we really want populations to be bigger than that. However, we also know that with Sarracenia alata, at least plants that are within a half meter of each other are clones. And so we really want to know in future studies, we want to look at more of those distances between selfing or half a meter and 35 meters. What's going on in those? Is there some inbreeding depression going on? Can those populations get smaller and withstand that pressure? And then we also want to look at the quality of offspring that's produced when we bring in pollen from other populations. Are they too distant to produce good offspring? Are we going to lose those localized advantages or not? And so we really want to pursue some more of those hand pollination treatments to see what the offspring look like from these pairings. Is there an interaction between genetics and nutrients, and how do they impact reproductive success in Sarracenia? Our data really shows that it's really important in Sarracenia that we're examining multiple years of resource restriction on individuals because they are storing so much of their nutrients. And we're not quite sure down the line how that's affecting them and how long they even store that assimilated nitrogen, how that fluctuates with other pressures. This is an experiment that's best done in a greenhouse. If you're unfamiliar with Sarracenia alata, I'll let you know that when they come out of dormancy in the spring, they don't always pop up pictures from the same growth point. They can actually pop up pictures from different areas of their rhizome. And so it's really hard to track plants that seem to be moving within a live environment. And so it'd be great to have these guys in pots in a greenhouse where we can really track these pairings and track these individuals over time. And lastly, how can we use this information to improve conservation of fragmented plant populations and especially Sarracenia? And while our experiment didn't show any effect of pollen donor distance on offspring, there is work done by Sheridan and Perot in the 90s, and they did work on Sarracenia flava, which showed that parentage is really, really important when you measure the pictures on their offspring. And so as often we see that you can't really measure reproductive success until you see the offspring or to see the offspring's reproductive success. And so really as land managers and enthusiasts and researchers, we need to be protecting these remnant bogs or these areas that we have, these habitats of Sarracenia for as long as possible so we can assess them long term, because we really can't take a snapshot of these bogs and understand their potential or any of the factors that might affect them over the long run. And so that is the summation of my thesis work. It was a great time spending time in a bog. I'm from California, so this was the first time I'd been in a Sarracenia bog, and I got to spend the whole summer there and watch the evolution, and it was just a beautiful experience. And I would love to take any questions that you guys have for me.
All right, wonderful, Hali'a. So we have a couple of questions, and thank you everyone who is participating live. If you have a question, feel free to type it into the box. So you talked about fragmented land patches. Do you know how important private land owners are compared to public lands regarding conservation? Where are, just like Sarracenia alata, for example, are they half and half? Are they mostly on private lands?
Right, yeah, so out here in Texas, like 90-something% of the land is privately owned. And so private landowners are very crucial to the success of these bogs. And it's really important that we have really good relationships with them so that they see the value in these bogs and want to do their part in protecting them, not selling them to, oil people or, draining their wetlands for some other type of development. So yeah, out here, it's very, very important that we have some type of public outreach or relationship with landowners so that they really understand what they have.
Did you personally talk to more than one landowner, or did your professor, your advisor, is he interacting with a lot of people?
Yeah, so when Dr. Horner got started into Sarracenia, he met like everyone out here that works on pitcher plants or knows where they are. And he's maintained the relationship with this particular landowner for at least, I want to say 12 years. And so that's been really great. The landowner regularly sends us like pictures of the bog, like, hey, burned it, or like, we'll ask him to run down there and be like, are the flowers like, are they popping yet or anything like that? And he'll like run down there, he doesn't really know what he's taking a picture of, but he'll take a picture of it for us and send it over or give us any kind of information that we ask. So he's been really great. I didn't really talk to them. We would just come onto their property and they knew we were there. The dogs would come run down and bark at us and then they would play and we just go about our work. And so it was really nice. A lot of people around here in Texas, like, don't like to be bothered. Like, even if you're on their land and they know you're there, they're like, don't talk to me. And so it was actually kind of nice. We just got to go in and do our work and get out.
So I live in Florida and I know a lot of private landowners are part of like different fire ecology groups, and they all kind of burn together based on what the scientists say. So
yeah,
there's good.
Yeah, and this landowner burns every 2 to 3 years his bog and surrounding land. I'm not sure that his neighbors do much of that, but this guy really keeps on track. That's really nice.
If a seed is viable but doesn't germinate in 30 days based on your test, what does that mean in terms of fitness? Is this a popular occurrence of them being viable but not germinating?
Yeah, so there's a lot of seeds that could be viable, but the germination conditions maybe just aren't ideal for that particular seed. We see that often in the greenhouse. You'll have a random seed like germinate way later than everyone else. And so that's why we did the viability test with the tetrazoline chloride. It tells us that that seed is viable in ideal germination conditions, which again can differ. But the fact that it wouldn't germinate on filter paper that's moist in 30 days doesn't say it's like less viable or anything. Actually, the staining that we got on those embryos was really nice and dark red, and that means there's a lot of live tissue in that seed. And so in better conditions, maybe in needed soil or needed higher humidity or something like that, that seed could have germinated. So yeah, that's how we corrected for choosing that germination treatment that we chose to do.
Yeah. So that was a question from Jane. She wanted you to further explain the viability test. So you did a great job. So is this chemical used by a hobbyist or not really?
Yeah, you can order it online. It's really easy to use. There's a lot of universities that have instructions on how to use it. It's used in industry when big seed collectors or folks that are especially interested in plants that are really hard to germinate or need a long dormancy period before they germinate. This allows them to see, you can like sample test a batch of your seeds and see if it's worth trying to germinate them or anything like that. And so you can test old seeds with it and see if they're still viable and see if it's worth planting them out. And so it's used in a lot of contexts, mostly in agriculture.
Once they're stained positive, as in they're viable, do you, or probably not you, but does a hobbyist keep them in that environment and wait, or do they do something?
Yeah, so to see the embryo on Sarracenia, you actually have to break open the seed coat to see the staining. And you have to actually scarify them before you introduce the chemical so that the chemical can get in past. If you ever looked at Sarracenia seed coats, they're actually really, really thick. If you look at a close-up picture of them or under a microscope. And so that's why you're sample testing them, because you don't want to destroy your whole harvest. You just want to batch test them, and you get an overall estimate of what your germination rate is going to look like. And then you should see that reflected in your actual germination. You try it out.
All right. Jane says regarding the fragmented lands and private owners, she said, indeed, Bay Beach Wildlife Sanctuary here in Wisconsin started out as private land. So the private people are very important. Okay, now if you guys have any more questions, feel free to type them in. I have 2 more questions about non-carnivorous plants.
Okay.
And I hope you can answer them.
Yeah.
Do you think non-carnivorous plants in nutrient-poor soils are using the same method of holding nutrients from previous years to produce their seeds?
That's really interesting. I'm trying to think of, well, within those environments, we get a lot of trees, which have extensive root systems. We get huge heather shrubs, again, big root systems. I'm not familiar with those taxa, those genuses in any way, but I wouldn't be surprised if they have some storage capability for hard years. That's generally what roots are for storage. How they utilize them year to year is going to be dependent on the species itself and other factors in the environment, but it's likely. There are a lot of bulbing kind of plants out there and stuff like that. And so that's probably a common strategy out in those environments.
All right, this is kind of a follow-up of that question. We say that carnivorous plants have their carnivory adaptations to live and reproduce in nutrient-poor soils. Now, you just said the roots and large root systems, but are you familiar with other adaptations that non-carnivorous plants have that are living in the same bog, the same swamp, the same environment, but that are maybe the same size or, similar root size? How do we justify that they're getting
that they're there surviving?
Yeah.
Well, I mean, there's a bunch of differences with plants. So in those environments, there's a lot of like quick growing grasses. So like a lot of annuals that are just gonna pump out whatever they can, and then they die back in the winter. And so that's their strategy. They just push out a ton of seeds each year. A lot of plants too in those environments are really good at tolerating high levels of toxins and things like that, or anything like that in that kind of soil, which is bog soils are really anoxic, so there's not a lot of oxygen. There are a lot of nutrients stored in the peat, but it's not readily available to those plants. And so some of them have relationships with fungus that are going to be able to help them reposition those nutrients out of the peat. But really what they're dealing with is the high amounts of water, the low amounts of oxygen. And so there's a lot of plants that have different adaptations to survive that kind of environment, and can deal with the low nutrients as well. So, yeah, there's so much diversity, plant diversity out in bogs, and there's just as many strategies for surviving them as well.
Well, it's always fascinating, and we want to thank you, Hali'a, for doing a wonderful webinar. And for those of you attending live, it's going to be posted on YouTube in a couple of days, and we encourage you to check out our other webinars. And thank you, everyone, for attending.
Thank you.
People in the comment section saying thank you, they enjoyed it, and have a great day. Bye.
Wonderful evening.
Thank you.
The International Carnivorous Plant Society wants you to be successful with your plants. We welcome growers just getting started all the way through professional scientists. We started an annual World Carnivorous Plant Day to celebrate these spectacular plants. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.